Great. Hi. My name is Amanda Hernan. I am a relatively new, I don't know how long I get to call myself new for, but I've been here less than a year, so relatively new research scientist at Nemours. I'm also an assistant professor affiliated with the Psych and Brain Sciences Department at the University of Delaware. And today I'm just going to talk really quickly about the INBRI Pilot Award project that I received funding for. The project is entitled A Role for Astrocytes in the Treatment of Cognitive Deficits After Early Life Seizures. So my lab is sort of broadly interested in understanding cognitive impairments and psychiatric comorbidities that occur in the context of pediatric epilepsy. So kids with epilepsy are often diagnosed with things like ADHD, autism. They also have learning and memory impairments. And so we're really broadly interested in understanding at sort of a neural network level and astrocyte network level what the underpinnings for that are and if we can find effective treatment strategies for those impairments specifically. So astrocytes are one of the largest cell populations in the brain. They play really crucial roles in normal development as well as the function of neurons. They're actively involved in things like neuronal signaling and plasticity, neurotransmitter uptake. They control neuronal synchronization and oscillations. They also maintain ionic homeostasis. They set up synapse development, and they're crucial for the brain's response to injury and maintaining the blood-brain barrier, which are two pathways that are really important in epilepsy in particular. And a number of studies have shown that astrocytes are damaged or their function is somehow altered in epilepsy. And so this is largely adult epilepsy, and again, so we're focused in pediatric epilepsy and trying to understand the intersection between neurodevelopment, epilepsy, and the astrocyte role in all of that. So we've previously shown that ACTH, which is an endogenous part of the HPA axis, is able to treat cognitive impairment in epilepsy. And we have shown that it has actions way beyond its systemic pathway, which is this melanocortin-2 receptor pathway. And in fact, it's acting through a subtype of neuropeptide receptors in the brain to improve outcome. So these neuropeptide receptors, the MC4Rs or melanocortin-4 subtype receptors, are located both on glia, including astrocytes, and neurons. So we're interested in understanding what the relative component or the relative role for the astrocytic MC4Rs and the neuronal MC4Rs is in this improved outcome we see with treatment with ACTH. So we hypothesize that early life seizures will lead to reactive astrocytosis, impaired expression of proteins involved in glutamate reuptake, and ionic buffering, and that this will manifest as impaired neuronal glial interactions, which will be recovered by ACTH, but only when the MC4Rs are expressed. So we took sort of a two-pronged approach to look at both static and dynamic measures of astrocyte function after early life seizures with and without the MC4R agonism. And so the first aim, we will explore reactivity phenotypes of the astrocyte populations in the brain and putative sort of static alterations and expression of proteins that are involved in sort of these key astrocytic pathways. So we're just taking a straight immunohistochemistry approach to look at the structure and the number of astrocytes, the relative reactivity of these astrocytes using a GFAP antibody. We'll be looking at glutamate clearance with by looking at the two glutamate transporters, GLT1 and GLAST. We'll also be looking at connexin 43, which is a protein that's involved, is a gap junction protein that's involved in astrocyte to astrocyte communication. We'll also be looking at aquaporin 4, which is a water channel that maintains kind of potassium homeostasis. So we'll be looking at these at time points after the seizures. So we have a recurrent early life seizure model that we're using, and we'll be looking at 2 days after the last of the seizures, what is happening to the state of the astrocytes with and without the treatment. sort of the thing that's near and dear to my heart is dynamics in the brain, and I've recently become interested in astrocyte dynamics in the brain and trying to understand how neurons are talking to astrocytes, how astrocytes are talking to astrocytes, and how astrocytes are talking to neurons. And so to do this, we're going to be using a calcium imaging approach, looking at both neuronal calcium transients and astrocytic calcium transients. The plan is to deconvolve the action potential vectors with the astrocyte calcium transients, and we'll model this using a generalized linear modeling approach. So this approach will allow us to quantify the effect of astrocyte calcium signaling on neuronal firing in a way that's unbiased and statistically robust to allow us to really quantify that interaction. So in conclusion, we know that drugs that act on the brain melanocortin receptors, specifically the MC4Rs, are really well tolerated in humans and may provide a powerful therapeutic approach to protect developing brains from the damage that's associated with epilepsy. So we have evidence that ACTH, which is a drug that's given clinically now to patients, but is also an endogenous melanocortin peptide, can prevent behavioral impairments in actually 3 different models, 2 rat and a mouse model of early life hyperexcitability or early life seizures, and that very recent data say that this is actually MC4R dependent in support of our hypothesis. These receptors are on astrocytes and neuronal populations where they have known neuroprotective functions on both cell types. So our interest, again, is in understanding the relative contributions of both of those cell types to the neuroprotective effect we see in our model. So understanding the mechanisms of action of the ACTH could, specifically as it relates to astrocytes, which are a relatively understudied population in the brain, especially in epilepsy, could elucidate more therapeutic targets in the future and hopefully have some broad applicability for not just epilepsy, but potentially also other neuropsychiatric, neurodevelopmental diseases as well. So with that, I would just like to thank everyone in my lab. So my graduate student at UD, Ahmed, he actually came with me from the University of Vermont, where I was before. My graduate student who's still at the University of Vermont, Trevor, our lab research assistant, our big lab group neuroscience coordinator, Pravin, and we have 2 very talented research associates who are working also in the group. And so these 3 folks are actually shared with the Scott Lab, who I work really closely with here at Nemours, and my other collaborator, who's actually a mathematician at Jackson Labs, who has helped me kind of spearhead a lot of the GLM work. That funding. And with that, I will take any questions.